My name is Giovanni Ciotti, I'm an Indologist, and I am the spokesperson of the Palm Leaf Manuscript Profiling Initiative. I have been coming to Puducherry since 2006 to study and do research on Sanskrit and Tamil literature and manuscripts. And it is now more or less 4 years that I have a very close collaboration with the French Institute of Puducherry. The French Institute hosts a fantastic collection of more than 8,000 palm leaf manuscripts that I have been studying for a very long time and that I would really like to know more from the point of view of natural sciences.
Hello, sir.
Hello, Giovanni, how are you? And this is a manuscript. I am editing this. It is a very nice manuscript.
Okay.
This text is about Atmartha Puja Paddhati, and which is one of the main aspects of Shaivism.
Sure.
And they are doing in the temples. There are two kind of puja, Atmartha and Parartha. Atmartha for the own sake. So he is doing puja. And for my edition, critical edition, if you can tell me from which area and which locality it has come, and if you can add to that, you can tell the year of the manuscript.
Sure. Now there is no column, there is no scribal colophon here.
There is name is mentioned.
The name is mentioned. Okay.
But
the
the
place
colophon
is
is
not
not
mentioned.
mentioned.
Okay.
If you can tell me, that will be
We can
good.
try and have a
Okay.
look at the material features and see, okay, if we can
Thank
match
you
it.
very much.
Okay, thank you.
Okay.
One of the challenges that comes with studying palm leaf manuscripts, in particular palm leaf manuscripts from this region, is that we have very large collections, but there are no records that tell us precisely where the manuscripts came from within this region.
This is the one kind of, this is the
Mistake?
yeah. No?
Yeah, possible.
Written.
Possible. Sometimes it
Wait, maybe this is 18th
so
century.
do you think this is the date of publication?
Incomplete. Incomplete. Anchavadum, Aravadum. This is also 4 and 5, 6. 4, 5, 6. This is incomplete or...
Knowing more about the geographical provenance of the manuscripts would help us a lot to know, for instance, who was reading what where, whether certain texts were only read in the north of the region or in the south of the region or everywhere across the region. And it's not that the information is not there, but it is not explicitly there, let's say. Maybe the material properties or features rather of these artifacts could reveal more about their origins. And for this, we turn to our colleagues from the natural sciences who may be able to help us establish the provenance of the manuscripts.
Yeah, my name is Professor Markus Fischer. I am, first of all, head of this Institute of Food Chemistry. Since 10 years, we are the Hamburg School of Food Science. And then I am the head of the Department of Chemistry and also the head of the research field Artifact Profiling in the Cluster of Excellence, Understanding Written Artifacts. Originally, we are a food chemistry lab. And normally, our people are going to analyze foods. But the technologies we use to analyze foods are the same which we can use to analyze other materials. So therefore, we can analyze those objects and just compare the profiles, the chemical profiles, like a human fingerprint. This is a very modern direction of investigation, but you need instruments. First of all, you need high-resolution instruments, and then you need lots of chemometrics and bioinformatics, because we have lots of data. The more data you get, the better is the picture. So in a perfect world, we would like to transport the artifacts to Hamburg and then analyze the artifacts in our so-called high-performance labs. The problem is that we have to get the permission to bring the artifacts out of India to Hamburg. And this might be a problem. And to make the story short, this was the idea of the container lab. So with the container solution, we can kill several birds with one stone. First of all, we are mobile. So this is very important. In short, the objects can remain at their original location and do not have to be moved to Hamburg. And second, with the container laboratories, we have a highly developed laboratory environment that meets our standards. So the first mission is India, but they will go to other places around the world. The container laboratories are the same, but the instrumentation will be different, depending on the question. So we have 5 laboratory containers, and we have one more, it's called a supply container. Half of the containers can be run under clean room conditions, and the other half of the container village, I call it container village, are just wet chemical laboratories. Yeah, the technical part of the container is the heart of the container.
And the heart of the container system is located on the rear side. I just open the door.
These are chemical laboratories, and there are several things which are very important. Temperature control is very important. The ventilation.
Condition, filter system, pumping system for fresh water and wastewater.
All instruments don't like high humidity. Therefore, we need a highly advanced ventilation system, air conditioning system, and this we have in our containers.
And here's the fresh air,
Coming out from
which is going here to the
I
filter
see,
and
okay.
here.
So most of the energy we need to run the containers are going into the air condition system. And the quality, for example, for electricity is not always given. That brought us to the idea, just we bring everything with us. So we are completely independent using those container facility.
Okay, you have this.
When the container arrived, we had already selected this warehouse, which was of course like completely bare. There was nothing, only the walls and the floor with a lot of holes and so forth, because it hasn't been used for many years. So what happened is that we leveled the ground, placed the containers, and then built everything you see all around it. Most of the retrofitting of the warehouse happened after the containers were already placed. So, for instance, the ground had to be leveled, the opening had to be closed with these metal shields, or doors, let's say. A water moat has been built around, and this is part of all the things that had to be done for pest control, because of course we are in a tropical environment, so there are insects and rodents that are very much interested in entering the lab themselves, and therefore the water moat is, for instance, useful to prevent the coming of all the insects that do not like water, so ants and scorpions. But at the same time, once you add water in this environment, you also add mosquitoes. So we added fish to the moat, little fish who will swim around happily, trying to catch and eat all the larvae of the mosquitoes. Of course, we also have mice, so we had to place some mice traps all around the facility, basically. And then you can see the stone chips, which are going to be all over the place. And these have 3 main functions. One is to prevent big snakes from coming, because if they crawl over the chips, they may get wounded, so they don't like it. The second is to prevent prevent sand from flying around too much because it gets like trapped and absorbed in among the chips. And the third one is when there will be the rainy season, it will also absorb more water, and so if we walk over it, shoes and boots will be less dirty once they approach the container, basically. And finally, the roof, which is a thatched roof made of, guess what? Palm, the fronds of palm trees, which is a traditional way of building roofs here in South India, let's say. And this has, of course, the function of protecting us from the sun, which is very intense, of course, here, and partly also from the rain. So if the rain is not like very heavy, simply the water will percolate on the sides and not over the container. Now we can test the containers in this particular setting and see if it works, because now with this kind of roof, we know how much energy we consume when the weather is very hot, but we have a good roof, and therefore how much energy we consume for the AC, for instance. These are all things that we are learning here, and hopefully we can apply them later when the containers will go for the future missions in similar environments or different environments. So the handling of the containers is something that we have to learn about and adapt every time we go to a different condition.
At IFP, there is a long tradition of working on the text, on the manuscript, but we never did any material analysis on the manuscript themselves because we don't have the team for that, we don't have the instrument, we don't have the capacity. And so that's what I found very interesting, is that I found that very complementary with the analysis of the text to be able to work on the materiality of the palm leaves themselves. From the beginning, one of the challenges is that part of the team was not sure that it would be a project where it will bring something to IFP. And so that's where the challenge is to be sure that people can be, for example, trained, or also to have access to the new data so that we can also work on them and not just give the manuscript and the logistic, but to be sure that there is an exchange and a real scientific collaboration with a publication done together so that the local team can understand more and be open to a new type of data. So that project also involved not only researcher and research institution, but also, for example, the German and the French consulates to get all the authorization. Also the government of Puducherry, who authorized us to use the port site. We are very thankful for that. What is also interesting, beside the interdisciplinary approach, is that it's also with a lot of different institutions, Germans, but also Indians and French from Puducherry, but also Bangalore, Hamburg, and that's always a challenge to work all together, but that's also where it's more fruitful.
Somehow, if I know my ladies, they want us to get inside and close the door. So here you go. Everybody in.
I'm Professor Sharda Srinivasan from National Institute of Advanced Studies in Bangalore, and I work in the area of archaeomaterials research using scientific analysis to understand how to fingerprint artifacts and characterize them. So a lot of my work has been on metal artifacts using XRF and mass spectroscopy and so on. So this project was also very interesting because you're looking at new materials like manuscripts which have not been studied from the Indian context. So there was a lot of excitement and interest in this particular project because a lot of times when it comes to archaeological material, there's of course been a lot of work on the inorganic materials, but organic materials, and I think particularly the case of manuscripts and manuscripts in India which haven't received that much of attention from the scientific perspective. So there's a lot of potential and it's very exciting for us.
Marina, this is a manuscript of the Pratishta Vidhi, and I would like very much to know something more about its provenance, if possible. So I will unwrap it for you and show you where there is the statement by the scribe, the colophon that says where it was copied, which is usually towards the end. Yes.
Oh, I see. Yeah.
Okay, you see here, it looks like a conclusion. Let me do it. Okay.
So we try to find as much information as possible. We start to take the samples at the IFP with a method which is non-invasive. So of course we won't destroy the manuscripts. More is always better. Okay.
Perfect.
And then we will bring it to the lab and analyze it. Then we carry out in the container the extraction process. And then we check the extraction protocol if this was successful. And for that, for example, we have a little sequencer here for check up the DNA extracts. Then we have the mass spectrometer here to analyze metabolites and peptides. And if the extraction protocol was successful, then we send the extracts to Hamburg, because there we have even bigger machines. And with that machine we can even gain more information. And then we will carry out a second measurement in Hamburg. And then at the end we hopefully can answer our different questions.
What the container lab does, it provides a controlled environment that is fully, so we do not outsource analysis, but we do everything within the team, and therefore all the parameters are constantly in check, and you can maintain them consistent, and therefore the results we get are, let's say, internally consistent. So that's why, even though, let's say, locally in India, for instance, there are, of course, labs that provide, DNA analysis, metabolomics, phytolith analysis, XRF, and all the techniques that we need to investigate this material. The fact of being able to do it within a confined lab space where we all work together continuously, let's say, and we integrate scholars and colleagues from the French Institute of Puducherry, for instance, but they become part of the same group. It's not like a separate lab that produces data and we just have to deal with the data. We produce the data together and we analyze and make sense of the data together. So within this controlled environment, controlled at large, let's say, then it is possible to really try to see something about these materials that make sense, let's say, and it is not just a patchwork, but it is pieces of the puzzle that we put together to create like a full picture. It has been the work of several years and of many people involved in the whole process to building the containers, testing the containers and deciding which equipment to put inside the containers, transporting them to India, setting them up in India, and finally coming to the point in which we can have the manuscripts finally analyzed. And of course, the selection of the manuscripts also has been the work of several years, let's say. And now I am extremely happy and as well as excited for the year and a half to come, more or less, where we'll finally be able to analyze the material features of the selected manuscript and be able to produce those data that, after some elaboration, let's say, will potentially tell us more about the geographical provenance of the manuscript, so that we can really step forward in our understanding of the transmission of the text within this region, and also as far as the craftsmanship that is behind the production of each individual item, because each individual manuscript has a story in itself to tell, and it is a piece of the past that has arrived into our hands and deserves to be studied in its own right. But of course, this is just the first mission of the Container Lab. This trip to Puducherry is the first one of hopefully many more that will come for the Container Lab. So the whole team is looking very much forward to the future missions and to see where the containers is going to go next.
This is not a one-mission project, so this is a sustainable laboratory environment which can be equipped with special instrumentation depending on the question we want to follow, and the container can be used many times. Will be used many times. They can go anywhere.